you in our broader community aware of who's doing what in the programs, where is leadership coming from, and what are some of our plans. And second, we want to get as much input as we can from participants because the UCSD stem cell programs are going through some strategic planning in the coming months. And also CIRM itself, as I'll tell you in a few minutes, is also undergoing strategic planning. And so it's really good for us to be able to collect ideas that might benefit one or the other of those programs. So I'm going to just share my screen right now and give you something that you may already know, which is the plan for the town hall this afternoon. I'm going to continue doing a brief introduction here. I'll then hand it over to Allison Moultrie and Sylvia Evans to talk about the stem cell program. I'll then tell you about the Sanford Consortium for Regenerative Medicine. Kat Jameson will then tell you about the Sanford Stem Cell Clinical Center. And then I'll give you an update on what's going on at CIRM and what are some new features of Proposition 14 that CIRM is trying to figure out how to best address. And then we'll have at least a half an hour for discussion with our colleagues on this Zoom call, primarily to get your input as well as to answer any questions you may have. So let's see here. Okay. Oops. So just to remind you, the way our stem cell programs are organized is pretty much, as I just summarized, the order with which each program will present its activities. And so Allison and Sylvia are the co-directors of the UCSD stem cell program. I'm the scientific director at the Sanford Consortium. More on that later. And then Kat, as I mentioned, is director of the Sanford Stem Cell Clinical Center. We have two excellent administrative people who help us, who can often find us even if you can't. Jake Banfield Ware, who's the assistant director of the UCSD Stem Cell Program and the Sanford Stem Cell Clinical Center, and Michelle Ghani, who's executive director of the Sanford Stem Cell Clinical Center. I then occupy two additional positions. One is as senior advisor for stem cell research and policy to the Vice Chancellor for Health Sciences. And I also serve on CIRM oversight, known as the ICOC, or Independent Citizens Oversight Committee. And I think that's all that I want to say about the general organization of the programs. Next up will be Sylvia Evans and Allison Moultrie, who will tell you about the UCSD stem cell program. So I've stopped sharing my screen. Allison and Sylvia, take it away.
Yeah, I think we decided that I'll talk, but Sylvia, feel free to interject. So yeah, we are both co-directors of the stem cell program. We are housed at the Sanford Consortium, and I think Larry will go over more about the consortium later on. And what we decided to do is just to highlight some of the facilities and activities that is being going on with the stem cell program. I will start with some of the cores, making sure that everybody knows about that. We do have a human embryonic stem cell core, and it's heavily focused on sorting cells, but we do have other expertise as well. This is directed by Paul Wheeler as well. We also have a stem cell genomic core that is directed by Jeannie. I think people should keep it unmuted. I think to avoid distraction, right? Unmuted. All right. Okay. So yeah, so we've been focusing more on single cell methodologies. Jean has been like a pioneer in this field and helping us all to navigate through that. We have also coming soon the electrophysiology core. So this is something that the community has been asked for a while. This core would help us to record from different cell types, and we have someone proposed to lead the core who already has agreed to come and serve for us. And this would be probably led by Martin Marsala if he accepted the position. All right, on science communication, we are doing different things. For example, I think we are all familiar with the Southern California Stem Cell seminar series hosted by Evan Snyder. This has been going on for, I think, over 10 years now. And we always try to favor cross-pollination by bringing outside speakers. Of course, we have to rebrand this during the pandemic. It's virtual now, and we are calling them breaking news on stem cells. We are asking people who have published recent papers to come and talk about these recent publications, and it's been like a huge success. And incredibly, all the speakers we asked so far to talk accepted. So another thing that we've done is a closer look where we invite someone from the basic science and someone from the clinical side and a moderator to talk about a specific topic with a more layman language. This is also has been very successful. We are recording all these episodes and making them available. So if you have ideas for that, I mean, we are welcome to work with you to propose one of these episodes in a specific topic. So we also have lots, we favor and help the implementation of local conferences. So some people from the community come to us asking support. We do have our major stem cell meeting. This year is going to happen between October 13 and 14. But we have other small meetings as well. For example, last year we had the stem cell ethics meeting, and for this year we have two proposed meetings, including one in glia cells for the folks more into the neurospace. We hosted the SUM 2020 grantee meeting, and again, I mean, if there is any interest from the community on a stem cell related topic, we can help to organize and implement those meetings. So on the teaching, we are participating on lots of teaching. I think the perhaps the stronger opportunity here is coming from CMM 250. This is also led by Carl Walling, where we have our winter courses here at the consortium. During the pandemic, this was all virtual, but these are nice opportunities for faculty to teach and also for students to learn about stem cell biology. On the outreach, we create a stem cell channel where we have lots of videos and layman explanations about different aspects of stem cells. It's housed in a YouTube channel, so it's freely available. Everybody can see it, and is a partnership with UC TV. There's lots of other activity. The stem cell program participate on faculty recruitment. Again, as I mentioned, conference sponsorship. We are also involved in equipment purchases if there is large interest from the faculty, and we have liaison communications with different institutes nearby or not as near, such as the San Diego Zoo. So we're also helping with grant support and preparation and submission. And that's for the other activities. Just to highlight some recent activities that's going on from the stem cell program, we participate on this NASA grant with lots of other folks to create to develop stem cells at the International Space Station. So this is a 3-year, $5 million grant that we recently got. There is a new center. We are calling them the Archaization Center. This is to study the impact of extinct genetic variants in human stem cell models. And finally, we are also participating with CENI, the Center for Engineered Natural Intelligence, in a project sponsored by Microsoft to create neural networks that are derived from organic networks coming from organoids and iPS-derived cells. So I think that's what I have. And if I understand correctly, the format, we're going to take questions at the end. Is that right, Larry?
Yes, that's exactly right. So,
all right, then I'll stop sharing here.
Great, thank you. All right, must be my cue to take over again. Allison, could you stop? Yeah, there we go. Screen sharing. Okay, so I want to pick up the theme and talk about the Sanford Consortium for Regenerative Medicine. I know folks in our community, some have more and some have less experience with it. So The Sanford Consortium was primarily set up as a laboratory building to give some spaces that were not federally supported so that we were insulated as much as possible from the political winds in stem cell and fetal tissue research in Washington, D.C. To do this and to make sure that we developed what was going to be and now is a multiple institutional building, we're set up as an independent 501c3, which means we have our own financial system and our own governance. And what we do is we lease space back to multiple La Jolla institutions, in this case, UCSD, Salk, Sanford Burnham Prebys, and La Jolla Institute for Immunology. And the building and the investigators that were brought in were all designed intentionally to enhance collaborations in stem cell biology and medicine as much as possible. And those of you who've been in the building have seen how the architecture actually promotes random interactions of people and faculty of different groups. And we, as in our experiences, leading to a variety of unexpected collaborations, as are some of our other activities. As I mentioned, we currently house investigators and cores from Sanford Burnham Prebys, La Jolla Institute for Immunology, UCSD, and Salk. And I'll just point out that the UCSD occupants have been very intentionally brought in so that they represent a wide range of different disciplines so that we're inherently interdisciplinary. And as you can see in the list here, we have faculty from bioengineering, biological sciences, medicine, peds, anesthesia, neuro, CMM, radiology, pharmacy and pharmacology, pathology, and reproductive medicine. So, and in fact, the collaborations that have been springing up around the building and between building residents and stem cell investigators across the Mesa are often interdisciplinary. We also house the core facilities that Allison just mentioned. And I'll just point out, we also have a very good high intensity MRI imaging center for rodents in addition to the other cores. All right. So in terms of how we're organized and paid for, as I mentioned, we do have an independent 501c3 status. Oh, it didn't change, sorry. There we go. And the initial financing that was done is shown here. We got a $43 million grant from CIRM as part of their facilities building program so that we had some insulation from the vagaries of the federal system. Denny Sanford very generously gave us $30 million, and we were able to finance the last $62 million with the help of our partner organizations. Our internal governance for the facility and faculty runs primarily through a scientific steering committee, which I chair. And of course, I also serve as scientific director. They're basically the same position. And the members of the committee are drawn from each participating organization. Anjana Rao from La Jolla Institute for Immunology, Marty Friedlander from Scripps, Evan Snyder from Sanford Burnham Prebys, Rusty Gage from Salk, and Andrew McCulloch from Bioengineering. Andrew has recently come on to replace Shu Chen, who was retiring. The Scientific Steering Committee also has three ex officio members, Ed Holmes, who's president and CEO of the consortium, Louis Kaufman, who's COO and CFO of the consortium, and Jean Yo, who's chair of our space committee. Not surprisingly, space is an important activity that we are frequently making decisions about. And the Scientific Steering Committee ultimately is responsible for determining the occupancy of the building, space allocations, recruiting, and miscellaneous procedures that we have dealt with administratively. And in the past year, of course, that's largely been COVID-19 testing for SARS-CoV-2 and keeping our cores operational. We also report to an independent oversight board, and that board is made up of the president or CEO or chancellors from each of the partner institutions, as well as the vice chancellor of health sciences, David Brenner. And then we have some of the usual suspect community members such as Denny Sanford himself and other folks who've been part of this over the years. Yes, so I think that's where I will stop and turn it over now to Kat to talk about the Sanford Stem Cell Clinical Center. So Kat, take it away.
All right. Thanks very much, Larry. So I just wanted to say, Larry and Allison and Sylvia and Jake, what a pleasure it's been to work together for the last several years, and what a nail biter it was for Prop 14, as we all know. I think I'm just now growing my nails back. So that was a $5.5 billion prop, as you know, very important, but they really are going to rely on their external advisors in terms of how they look at the future 10 years of funding, with 1.5 billion of that being set aside for neurological diseases. So in terms of the Sanford Stem Cell Clinical Center, we started in 2013 with a $100 million grant from T. Denny Sanford to really put our money where, or put his money where our mouth is, and that is translate the discoveries from the bench to the bedside, which is easier to say than to actually do. So we realized that we needed to have an infrastructure to do that. And that included putting in physical space and also being able to hire the appropriate faculty and staff that would help us to initiate clinical trials, but perhaps more importantly, do pre-IND and IND enabling work that would get us there. These are the things that usually only happen in industry, and it's really pretty extraordinary that we've been able to do this a few times here. We started 3 first-in-human clinical trials in September 2014. One was the neural stem study, another one was the ViaCyte study for diabetes, and then another one was sermtuzumab for cancer. So these linked organizations, the Sanford Consortium for Regenerative Medicine, the Advanced Cell Therapy Lab, the Jacobs Medical Center, Sanford Stem Cell Clinical Center Inpatient Unit, and the Komen Family Outpatient Pavilion, where we have the Sanford Stem Cell Center, where we do outpatient work, really all linked together. And this has been an incredibly important really structure for us to be able to get your ideas from the bench to the bedside, and has been really pretty remarkably successful. So in terms of what do we do at the Sanford Center, we do have some pass-through funding to the stem cell program. We help to coordinate the conferences like the CIRM Grantee meeting and the Sanford Center yearly symposium that some of you will be speaking at, and Larry gave the keynote for last year. But what we really want to be able to do is take the ideas, make them clinical realities by having a pull-through program where we look to see what nascent ideas we should be helping to partner on. We have regulatory resources, and we're expanding that team with Vicki Scheckler leading the team. So we'll be able to help with pre-IND and IND enabling studies. We do provide some counseling. We'd like to expand on this to include multi-omics navigation, and are discussing that now. Clinical trials, we have a broad range of clinical trials that include small molecules, biologics, and cellular therapeutics to enhance endogenous repair and regeneration, or just to replace degenerated tissue. We have genomics and imaging. We'll be doing more molecular diagnostics in the future, and you'll see that we'll be using very specific PET imaging probes. And of course, we have, in addition to whole genome sequencing, whole exome sequencing, we're trying to bring RNA sequencing into the clinic, as well as proteomics, usually by a phosphoflow strategy. In terms of clinical care, we have a division of regenerative medicine that we started in 2014. I'm the inaugural chief of that division through the Department of Medicine. We had the capacity to hire FTE faculty to this division and have an inpatient cell and regenerative medicine service where we see people who have cellular therapy needs or stem cell related clinical care needs. We also have the outpatient center, as I mentioned, and there we can see patients not only who are undergoing clinical trials, but have the need for standard of care targeting cancer stem cells, need for neurological regeneration, or diabetes, and we'll expand that into musculoskeletal disorders. So this has really grown. So Larry Goldstein was the initial director of the Sanford Stem Cell Clinical Center. I took over just about a year and a half ago, but we still work together very closely and also work very closely with Sylvia and with Allison, so we can really interlink our programs. So what you see is we have pull through from the Sanford Consortium and UC Stem Cell Program. And really the point of this webinar is really to try and expand this network to include more of our faculty members on the Mesa. And so we can really bring to bear the major strengths on this campus. In addition, you see from a genomic standpoint, we work very closely with Jean Yeo, Louise Laurent, and others with the COVID crisis. You've seen how important their skills have been for actually being able to test all of our students who come into the building and our researchers. We have the Advanced Cell Therapy Lab. This is run by Dan Kaufman, who was recruited to the Division of Regenerative Medicine just about 4 years ago now, and that has really bolstered scientific collaborations. I mentioned the Jacobs Medical Center, the Komen Family Outpatient Pavilion, and perhaps more importantly for clinical trials, we have a CIRM Alpha Stem Cell Clinic. This is a $16 million grant, $8 million from CIRM, $8 million from our center. It had to be a match. And we've been able to bolster that clinical trials infrastructure with Sanford Stem Cell Clinical Center funding. As you saw from Prop 14, and Larry, I know you're going to go into this a little bit more, the mandate from Prop 14 is to expand the Alpha Stem Cell Clinic network. And so that's a very important initiative. They also plan to have community care centers of excellence. So what are alpha clinics? So these were started, Alan Trounson was the chair of CIRM, and he wanted to make sure that we could take these stem cell ideas, these major discoveries, and have them implemented clinically in a very streamlined way where we would be able to share the technology across these centers. There were initially 3 alpha stem cell clinics funded in 2015, and then we added UCSF and UC Davis. So all told, the Alpha Stem Cell Clinic Network actually reaches 20 million Californians. So it's quite a lot. I expect that this Alpha Clinic Network will expand. You see the centers here, City of Hope, UC Davis, UCLA and UCI form one center, UCSF, and we have a center here as well that I direct. We are able to use recruitment tools and registries in this network. We have a single IRB reviewing for multiple sites. We implemented an accelerated confidentiality agreement and clinical trial agreement platform, and we are able to partner throughout the network for some really pretty involved clinical trials. So we bring to bear some stem cell therapy expertise, the actual medical centers in the network. We have an operational support team on site, and we are really involved in patient education and trying to really foster the idea that patients can ask questions. And, you know, as we mentioned, or Allison mentioned, we have the Closer Look seminar series. I frequently find that patients are watching those. So patients are just much more savvy these days. Just to give you an idea of the portfolio, this is going a little way back, but what Larry and I had first envisioned when we wrote this grant together was that we would go from phase 1, where it's basically safety, tolerability, and proof of concept, to phase 2, where you say, are we at the right dose? Phase 3, is this better than standard of care? So we've actually managed to do all 3 phases within the Alpha Stem Cell Clinic, and this is just expanding from cancer and neurodegenerative disease, as well as diabetes, to some rare diseases. So one of the surprises for me was that one of the best accruing studies is actually Stephanie Cherky's clinical trial for cystinosis, which is rather a rare disease. But it's a stem cell gene therapy trial. There's no other treatment that's effective. And so that one has accrued very well. We provide regulatory support. So the point of this slide is we don't have to provide support for everything in the clinical trial. We can actually provide support for just parts of it. We're doing trials for cardiovascular disease, renal disease, inflammatory bowel disease, and osteoarthritis. So we expect the indications to expand. And as you can see, we're transitioning nicely from one phase to the other, with an example of that being Sermtuzumab. Sermtuzumab is an antibody we developed locally with CIRM funding, so we called it Sermtuzumab. Tom Kipps led the team for development. Dennis Carson and I were the actual PI and co-PI of the CIRM Halt Leukemia grant. And this is now in late phase 2 studies for CLL and mantle cell lymphoma, and now has expanded to include breast cancer. And the breast cancer data actually look very exciting. So those will be presented at AACR. So this embryonic antigen that's re-expressed by a large number of malignancies as they invade and metastasize looks to be very readily targeted by this antibody targeting ROR1. Neuralstem, Dysgenics, these are all clinical trials for people that have either degenerative disc disease or spinal cord injury. We're going to add 2 more trials to that. And then diabetes, ViaCyte, they're making a new technology. So we'll be moving on to that. In terms of the Alpha Clinic trials that we're doing, we're going to expand our current portfolio within the context of what we've had already. We've had 3 areas: oncology, metabolic disease, and neurological disease. And that's because those are the areas where we have the greatest stem cell expertise, where the stem cell field really does have a lot of knowledge with cancer stem cells, neural stem cells, and then understanding pancreas and related metabolic disorders like diabetes. So you can see we've got a good range. Most of our clinical trials are actually phase 1, and that's really where we want to stay. We really want to stay with this investigator-initiated portfolio. Enrollment for our center has actually been very good, and this has a lot to do with patient education. And Betty Cabrera, who has been leading our Alpha Clinics from a managerial standpoint, and then Sheldon Morris is the deputy director. So this all goes hand in glove with our advanced cell therapy lab that Dan Kaufman runs. This is a specialized unit and is able to work with a number of groups, including outside the university. So the ACTL works with industry sponsors. It has established a quality management system that complies with good manufacturing practices for drugs, biologics, human cell and tissue engineering. And, you know, we actually did the first product of tumor infiltrating lymphocytes recently, and the patient was treated on our Sanford Center inpatient service. The ACTL enables the effective and efficient use of resources. And what we're finding now is more people from industry really want to avail themselves of this specialized center. This is where we are, just to give you a 30,000-foot view. We've got the Sanford Stem Cell Clinical Center Advanced Cell Therapy Lab, which is just off campus Point Drive. And then we've got the Komen Outpatient Pavilion, as well as the Jacobs Medical Center, where we have the inpatient services. And then we've got our ACTRI offices as well for our regulatory work. So the layout is quite good. We would need to expand this in the future, particularly if CIRM decides they want us to do more of the GMP manufacturing for cellular therapies. And that's really it for me. I just wanted to be available for questions and just wanted to say what a privilege it is to work with all of you. I see Gabby is here. He's helped to recruit most of our faculty as the head of the DRM search committee. So thank you, Gabby. You see that we've got a lot of people here. One person who's going to give our keynote at the end of this year's Sanford Center Symposium, Don Cleveland, I saw earlier. Martin has run our Sanford Surgical Training Center, and I think we'll continue to do that, Martin, right? So there have been no serious adverse events, amazingly, in that neural stem cell clinical trial, because Martin Marcella and our phenomenal surgeon, Joseph Chachi, work together to practice the surgery before it ever goes into a patient. I don't know any other center that has that. I think we're still the only ones, aren't we, Martin?
Yeah, I think so. You're right. Yeah. And we'll continue with the next trial now with the lineage. Yeah. So we're looking forward. Yeah.
That's phenomenal. So that's really it for me. I just wanted to say what a joy it's been to work with Larry and the team, and looking forward to another 10 years.
Great. Thank you very much, Kat. All right, so let me take the screen back. So I just want to give a very brief overview of what's in Proposition 14, which passed with a little over 51% of the vote in November. It was a bit of a nail biter, but we're all quite happy that it ultimately passed. As Kat mentioned, overall, it's $5.5 billion to continue the California Institute for Regenerative Medicine and its efforts to develop high-quality stem cell-related therapies to people with disease. Prop 14 established a number of new programs, some of which the board and the staff of CIRM are now working to figure out how to best integrate. They're not all straightforward. One of the new programs includes establishment of what's called an accessibility and affordability working group to be sure that patients from diverse parts of the state can afford to be in clinical trials or can afford approved therapies once they develop. Prop 14 also sets aside $1.5 billion for research involving diseases of the nervous system or injuries to the nervous system. I'm sure many of you will have questions about what exactly that means. As Kat mentioned, it's going to expand the alpha stem cell clinics around the state. That network has been incredibly important for the development of stem cell clinical trials around the state. And in a new activity, is going to reach into underserved communities and create community care centers of excellence where patients in underserved areas can participate in clinical trials of the sort that Kat recently mentioned. Prop 14 reestablishes the training and fellowship programs, which I'm sure everybody is very happy about. That's moving forward very nicely. And there will be some sort of shared labs program, as well as facilities funding for the community care centers and the shared labs, although it's not as much as was set aside in Prop 71. I've just mentioned accessibility and affordability. It seems it is, in fact, a very important change in direction for these efforts by being aggressive in outreach efforts. And then genetic research is part of the wording of Prop 14. And I think what I can tell you at the moment is the board and CIRM permanent staff are discussing how best to interpret that. There's authorization for the board to make awards across the research spectrum, including awards for therapy delivery, while trying to retain, at least as its primary mission, addressing areas that are inadequately or politically dealt with by the federal government, such as embryonic stem cells, fetal tissue and fetal stem cells, and related materials. In terms of the dollars, almost all of the $5.5 billion, $4.7 billion to be exact, will go to research grants. Up to about 100 million will go to accessibility and affordability, another 78 for community care centers of excellent facilities, and then, as I mentioned, about 26 million for shared lab facilities. So not nearly as much as in the prior proposition, but of course, most of the institutions with major programs built major facilities in the first round. So in principle, we shouldn't need as much as we did beginning things. So what's next for CIRM? So what I can tell you is that CIRM is going through a strategic planning process for the next 4 to 6 months. The board is working with CIRM staff to try to figure out what are, the primary directives going to be and how are we best going to meet the needs of the state in the development of stem cell related therapies. In parallel with the CIRM strategic planning effort, Kat, Allison, Sylvia, and I, with support from Jake and Michelle, are beginning a UCSD stem cell strategic planning process in parallel, where I think we will make some decisions about major areas we might want to invest in, and also how to best support our faculty in the development of their programs and research grants. And I'll just close by mentioning Kat, Sylvia, Allison, and I will be presenting some of the semi-final ideas for our strategic planning process at the Research Council in May. And I know that has a very broad invitation and distribution list. So I think that's about it for the issues I wanted to cover. And I think this would be a good time to open things up for questions and suggestions. Jake, do you want to handle that by using the chat function or by people raising their hand?
I would say whichever one you're more comfortable with. Chat function's fine.
I was going to go the other way and say,
Oh, then let's do it like that. Raise a hand.
so if everybody, if you go to the reaction button on the lower part of your screen and click on it, you'll see that there's a raise hand function. I've just done it so you can see what it looks like. And then the software will queue people up in the approximate order of the questions, of their hand raising. So there we go. Sue.
Hi, so I had a couple questions and comments. It'd be great if a comment first is you probably realize that the School of Medicine is also undergoing a strategic planning process, and I would love to coordinate with you all. So I think it's really important. The other question I had is in terms of research grants, that I know you're undergoing the statewide strategic plan. Is your feeling going to be that there's going to be monies for studying stem cells in a non-directly therapeutic situation?
Yeah, so that will be a lively debate moving forward. I don't really know how it will come out. I'll tell you that my personal preference is that we want to continue studying the basic science of stem cells and pathways that stem cells use to generate tissues normally. Partly because that's a tremendous therapeutic benefit ultimately, is my view and many others, and partly because we need to expand the discovery base if this field is going to continue to grow and prosper. And I think it's a very valuable field. I think the citizens of California have recognized it, but as you know, the devil will be in the details.
Yeah, the other thing is, Susan, so the CIRM had an external advisors meeting, and they'll have a number of these meetings actually with people who specialize in very specific areas. So I was actually asked to speak about cancer stem cells in the alpha clinics. There's still a lot of interest in the basic science regarding the stem cell properties of cancer, but also potentially in centers of excellence and new initiatives entirely, as Allison alluded to. We have this $5 million grant with NASA and Space Tango. CIRM may be interested in reinvigorating another set of RFAs for that. So I'll be having a chat with the head of the ISS National Labs, Ken Shields, and Maria Milan coming up soon. So they may be wanting to look into some very interesting areas. But the external advisors are really adept scientists, and a lot of them basic scientists. So I think that we can look forward to having basic science funding, but Larry can give us the lowdown after the ICOC meeting, but it's certainly clear that they're looking at scientific stakeholders for input.
I'll just add that I'm hoping that the board recognizes something that I think is really important, which is the people of California ultimately really would like to see therapies developed. But I think they'd also settle for some biotech companies that develop therapies as well. And that's been an early theme where CIRM funding has led to a number of companies being established that have become quite successful. Kat's company, was it Impact, the first one, Kat? And then of course, Irv Weissman's company 47 have been incredible successes that will deliver good therapies as well as economic benefit to the state.
Okay, and just one more thing to follow up on. I'm very excited that the word genetics is being approached. And I guess the same question comes up as you don't know the context yet, really, because it could be how genes affect stem cell, proliferation, function, etc., or it could be more therapeutic genetic models and treating with stem cells, or a combination. But the details aren't out yet, or?
So the board has started to struggle with this question along with the staff. The language of Prop 14 doesn't put many limits on what's meant by genetic research. And so I think ultimately the board and the agency are going to have to figure this out. Again, my personal bias is I would like to see it at least related to stem cell biology, medicine, or regenerative medicine. The same goes for the gene therapy and neuroscience mandates. Personally, I think it would be a mistake for us to become a mini NIH. We really want to try to do things that the NIH won't necessarily do or won't do at the scale that we think is most appropriate for the state. So, but we'll see.
Okay, thank you.
There is a CERM Genomics Initiative, Susan, that is run out of UCSC. So that already exists. And so there is a bit of a template.
Yeah, I guess I'm thinking of genetics and genomics a little bit differently
Sure.
here. I mean, they're intertwined, but yes, thank you.
So if you're done, Sue, could you unraise your hand? Mana.
Thank you. Kind of along those same lines, I was wondering if there are any opportunities you think, Larry, through the new funding available through CERM for banking of well-characterized iPS cells. Thanks to you guys and through our new Center for Perinatal Discovery, I've been talking to several people in the building, including Evan Snyder, where we actually have a grant right now that just started with Jack Bowie and also Dan Kaufman to bank cells, matched maternal and placental cells from our newborns at UCSD. For the grant that we have, it's for looking at sort of interactions between placental cells and maternal cells at the maternal fetal interface. But these are going to be all from pregnancies and from neonates that are going to be very well clinically characterized. And potentially also with Evan, we've discussed how we would do whole genome sequencing as funds allow. And so the question is, if this bank is going to be available to researchers, at least at UCSD and beyond, but is there ways that we could find funding to maintain it and expand it?
Again, a great question. I don't know what the CERM posture is going to be with respect to cell banking. I mean, they've set up a large iPS bank for a variety of different conditions based on biopsies or blood samples taken from folks around the state. I guess, the first question would be whether it's possible to dovetail in some way with that effort as part of your wanting to make materials broadly available. I think that's another one of those watch this space kind of answers. I don't have a good answer for you at the moment, but obviously what you describe has significant merit and we ought to try to find a way to do it. I mean, the problem that the board and the staff are going to face is there's going to be far more important and interesting things to do than even $5.5 billion would accommodate, is my guess. So, some priority decisions are going to have to get made down the line. And that's going to be a large part of how this process plays out, I think. Thank you, Mana. Stephanie.
I unmute, sorry. Hi, everybody. So I know that CERM has been all about stem cells for many years and after included gene therapy. So there are a lot of gene therapy stem cell clinical trial. But I know they were also talking about using in vivo gene therapy with AAV. Do you know, my first question is, what is the status of that? Are they going to do that? So this is not including stem cells. And I also want to add, because we are really making an effort with UCSD Rabies to develop gene therapy, in vivo and ex vivo gene therapy. So, and that's why it would be important to know, because to see how we can include CERM into all this effort.
Well, I think it's going to be 6 months before I can give you an answer to that, Stephanie. I mean, it's a great question. And I think this is exactly one of the issues the board is going to struggle with, which is, should all of its gene therapy programs leverage off stem cells or progenitor cells in some way, or are they prepared to enter the field of gene therapy more broadly, which of course is a much bigger field, which, in some people's view, you might conclude that the NIH is doing an adequate job. And there's not so, okay, Stephanie disagrees with that. Okay.
No, I mean, it's not, but I mean, we see how we need much more money. I mean, NIH doing an adequate job for the basic science, but not going from bench to bedside. And CIRM is doing a much better job on that. So there's nothing that can compete with CIRM.
Yeah. So let me just mention something that you folks probably know but forgot. All of the ICOC meetings are open to the public. And so they either broadcast to YouTube, I think, or you can call into the meetings and listen to them. And there's a part of the meeting where individuals can make comments up to 3 minutes. And so certainly what I've heard at these meetings over the years are investigators such as yourself showing up to make a suggestion about something CIRM should consider doing. And of course, you can also write to the chair of the ICOC and the president of the agency. They're small, they're located in the state of California, they're very accessible. And so, obviously you're telling me about it is one way to get input into the ICOC, but another way of doing it is to, show up the meetings, make comments, send emails to the relevant people and make the case that this is something that CIRM can do uniquely, that other agencies are not adequately funded.
Yeah, I think another thing, Stephanie, that we've been talking about with CIRM, or at least I've been talking about with CIRM, is the need for a patient registry and for us to work as a network through the Alpha Clinic networks to look for adverse events. That is what we worry about with a lot of stem cell related therapies, including stem cell gene therapy, including AAV vectors. Although they're thought to be quite safe. We saw with the lentiglobin study for sickle cell anemia, 2 patients developed AML, acute myeloid leukemia. That's something that I study that affects 4 per 100,000 people, and usually people in their mid-60s, not people 50 and under. So the Bluebird Bio was put on clinical hold because of that. So I think that we have to really be careful what we wish for, in a sense. We have to remember that gene therapy has a lot of potential.
Yeah, I mean, on that, so CIRM did a really good job because we had a round table with 30 investigators involved in ex vivo gene therapy. And we came up with, I mean, it seems like it's ruled out the gene therapy. And we will come up with actually statements that will be used for all of us in our consent informants and public. So CIRM has been a really good addressing that.
So I think CIRM can safeguard the public essentially by insisting that we share our data, and we have a central data repository for these trials. And so it isn't just one-offs. I think the more we link with the network, the better. We can keep each other honest. And that's actually what's happened with the BMT Clinical Trials Network, with the CIBMTR. Usually for advanced cell therapies, you have FACT accreditation. So what's coming next is we'll have a fellowship training program for clinical fellows as well to be able to do regenerative medicine. So I think that by having CIRM, we'll be able to streamline these activities and make sure that they're safer for patients, and we have a registry and we can look for outcomes just like we do for blood and marrow transplantation. So that's what I see coming in the future, but they would need to put that in place if they're really going to put stem cell gene therapy forward as a major initiative. So I think they will, but they'll expand the CIRM Genomics Initiative as well to keep in step with that. Or that's what I'm anticipating, just based on the discussions we've had so far.
Thank you, Stephanie. Dan.
Thank you.
Yeah, hi. So sorry, I'll get on here. No, this is really useful. So a couple things. So one, I just want to follow up what Kat presented about the ACTL, our GMP cell and gene therapy. And so there is a workshop with CIRM in a couple weeks that they've called for a select group. It's sort of invitation only, where they're going to discuss plans presumably to expand this. So this is a great selection of people. If you have projects, that you may be looking for GMP cell or potentially gene manufacturing, not just now, but thinking 3 to 5 years down the road, let me know. Just send me an email, and I'd love to get an inventory of what's in the potential pipeline. We're already working with a number of folks, and again, as Kat mentioned, this can be both internal projects. They've done work for my lab, work with Ezra Cohen, Greg Daniels at the Cancer Center, spin outs from UCSD. So we're working with a group doing iPS drive retinal cells. And then we're, also actually now the lead manufacturing site for actually a Stanford spun out biotech BioEclipse, where they chose to work for us instead of Stanford. So we're actually now treating patients. This is a cytokine activated killer cell population. So if you have ideas, let me know. If you need something sooner, go to our website, actl.ucsd.edu, and there is a page to fill out an inventory form, or a project proposal for what you may be interested. But it's great to have everybody here and to bring this up. I do have a totally unrelated question for Larry. So, this area of community engagement and these community care centers, which is obviously not something I work with, but I'm sure there's others here who have interactions with this. My wife is actually a family physician with Family Health Centers of San Diego, right? And this is, her clinics in City Heights. This is not a group that is looking for, I'll say high-tech interventions. They need insulin, right? Very basic care. And so how CIRM is gonna, bridge that gap and to engage them, they, don't necessarily need, clinical trials for a ViaCyte device, but they need, access to first basic medical care and then, potential trials. But this is a very, I think the challenges for this type of underserved population is, obviously very different than what we have here in La Jolla. And are they going to have some community engagement, or what is going to be the process to fulfill that goal?
Yeah, so I mean, I can give you a partial answer. So the ballot proposition defined a working group in affordability and accessibility, and that working group is currently being selected right now. They're looking for people with expertise in delivery of care into underserved communities, healthcare executives, clinical trialists, and I think it's an open question about, what exactly is going to be needed in those communities. I could well imagine that participating in a cancer trial with Sirmtuzumab might be something that some members of those communities might find important if they have the right kinds of cancers. So, I think we're going to have to see, Dan. You're right. It's a new project. We don't know exactly how it's going to be organized. But I think, the board and CIRM will do the best they can to meet that challenge.
Okay. So again, last unrelated question, but as Mana brought up, so the existing IPS banks that CIRM has funded, if I was interested in identifying IPS cells with a specific genetic characterization, right? What information is available from those banks? Do we have genome sequencing if I'm interested in a specific SNP or specific, HLA haplotype or what? Who has access to that data?
I think my recollection is that there's a web link that goes to the bank and the cells, and I think ought to have information about what the characterization is. But I'll say, as far as I know, they have not been subject to large-scale sequencing. Kat or Jean, do you know any different?
Yeah, so they do. My thought, having talked to Maria Milan, the CEO of CIRM, is that they really want to enhance the use of the CIRM Genomics Initiative and have the equivalent of dbGaP, where the data would be deposited centrally at a CIRM site, and then we would use the CIRM Genome Center to do things, the exact things that you're suggesting. Whether they're paid for as a separate initiative for genome sequencing of IPS, whether it be whole genome or whole exome, isn't clear. But I got the impression they really want to expand that. And the IPS cells were a main area of interest for the external advisors when I saw the external advisor report. And you'll get an idea of that too, Dan, when you participate in that meeting with the external advisor. So you'll have access to the report. And then Larry will be part of the ICOC to see what the priorities are going to be. But it would make a lot of sense to do sequencing, especially now that it's become so cheap to do whole genome sequencing. And it certainly would make sense for stem cell gene therapy products as well.
Yeah, no, that's a really good resource that's already, it's like, 80% of the work's already done. So getting that sequence would be quite valuable. So, okay, thank you.
Thanks, Dan. Bob Hevner. Bob Hevner, your hand is up.
Hi, Larry and everyone. Thank you so much for this wonderful, informative presentation. I've really learned a lot. My question has to do with the funds from the newly approved proposal. As I remember from the last iteration of the CIRM grant, there were different tracks for grants to investigators of sort of basic science and then more clinically oriented. Am I to understand that that's still not worked out yet? That's still under discussion, what's going to be available for investigators that we can apply for, or do we know that at this point?
Yeah, so I think it's fair to say that that's part of what the strategic plan is going to develop, is perhaps, I hope it would be explicit enough to give us some target allocations for different classes of investigation. I mean, if you look at the CIRM grants page right now, there are 3 types of grants that are currently available, which are actually a holdover from the last round of funding from Prop 71. And they're a basic science discovery type grant, a translational grant, and an early clinical trials grant that investigators can apply for. One thing I'll note is that leading up to the election last year, CIRM was in a close down phase. They didn't know they were going to be refinanced. So a lot of people left, a lot of programs were terminated, and they went into survival mode to be sure that they could adequately fund commitments that they'd already made in case the ballot proposition didn't pass. Because although there was early optimism, the COVID crisis really blew a hole through the side of the campaign, and it's a wonder that it did as well as it did, to be honest. So it was quite a feat. So I think this is going to be something that emerges, is some idea of what kind of allocations for what we would call basic stem cell science, recognizing that basic science is an important part of therapy development. It leads to new ideas and new approaches, and I think has to be part of the portfolio. But there has to be a clinical part as well, hopefully earlier than we did in the last round, but a lot of the early development work has already been done. So, stay tuned. Okay, thank you, Bob. Who else is next? Bob is, I see. Yes, great. So I'm not seeing any hands up or anybody who has a burning question. Everybody on this call knows how to reach any of us by email these days quite readily. And I would encourage interactions with CIRM directly. The agency is there for all of the people of California, including the scientists and clinicians on this Zoom meeting who would like to use those funds to do things that are beneficial to the state. So please participate directly. Let us know your ideas, and we will do our best to run these programs effectively. So if there's no further input, I'm going to declare us adjourned. Thank you all.
Thank you. Bye-bye.
Thanks, everyone. Have a great day.